Hello everyone, welcome to this Frontiers in Science deep dive webinar on NANOSPRESSO toward personalized, locally produced nucleic acid nanomedicines. I'm Lee Baker, the Editorial Adviser at Frontiers in Science, and I'll be your moderator today. Before we get into today's topic, just a few words about the journal Frontiers in Science. This is our flagship multidisciplinary open access title at Frontiers, focused on transformational science for healthy lives on a healthy planet. We commission exceptional peer-reviewed lead articles from world-leading researchers, and then around each of these articles, we build a unique hub of multi-audience content, including scientific viewpoints, editorials, policy articles, an explainer for the general public, even an article for kids, and a webinar such as the one today to make sure these articles get maximal reach and impact across scientific audiences, but also policy and the general public. It's all designed to make sure that transformational open science reaches and benefits everyone. So today, we're focusing on this lead article titled Toward Personalized, Locally Produced Nucleic Acid Nanomedicines, which we just published just last month. It was led by Professor Ray Schiffelers from UMC Utrecht with a multidisciplinary panel of co-authors, including Professor Sabine Fuchs and Dr Alexandre Ceccaldi, all of whom are with us today. As I'm sure you'll know, nucleic acid therapeutics could offer precision medicine interventions in a variety of conditions, including rare inherited diseases and cancers and in new vaccines. However, these therapies are currently held back by many obstacles, including pharmaceutical manufacturing challenges and costs. This article presents the pioneering NANOSPRESSO project, which fuses nucleic acid therapeutics and decentralized nanomedicine delivery platforms to provide a model for localized production of precision therapies at the point of care, potentially offering benefits of agility and affordability and convenience. Here's the full article. And as always, it's published within Hub. This is the Hub, which is together. We have an editorial by Professor Christoph Klein and Daniel Petersham from the Ludwig Maximilian University and German Center for Adolescent and Child Health in Munich. We have viewpoint articles by the peer reviewers, Horatio Cabral from the University of Tokyo, Klaus Wallstadt, and Olga Kokova at the University of Miami. And we also have a policy outlook piece by Alexandra Heumber and Deborah Bellon from Rare Disease International, and Alexandra joins us here today. You'll also find a lay explainer, infographics, and a video, and as I mentioned, an article in Frontiers for Young Minds, our journal for kids. So today's program is here. First of all, Professor Raymond Schiffelers from the University Medical Center Utrecht will open the show to explain, set the scene for us about the NANOSPRESSO concept. Professor Sabine Fuchs will then take forward the applications of gene editing therapeutics delivered by nanomedicine therapeutics for metabolic diseases. And then Olivia Lewis, also from Utrecht University and involved in the NANOSPRESSO project, will dive deeper into the regulatory issues surrounding the use of this new program. We'll then have a panel discussion in which we'll also include two additional panelists, Dr. Alexandre Ceccaldi from the European Technology Platform on Nanomedicine and Alexandra Heumber-Perry from Rare Diseases International. So first up, I'm delighted to welcome Professor Raymond Schiffelers, Professor of Nanomedicine at the University Medical Center, UMC Utrecht, and leader of the NANOSPRESSO project. Ray's a leader in nanoparticle-based systems for precision drug delivery. He's a key figure in several European consortia, a founding member of the International Society for Extracellular Vesicles, and the immediate past chair of the European Technology Platform on Nanomedicine, the ETPN. Recently, Raymond received the prestigious Advanced Grant from the European Research Council for his research on smart nanoparticles for cancer therapies. So thanks, Ray, for bringing this transformational project to Frontiers in Science, and the floor is yours.
Well, thank you very much, Lee. Thanks for hosting us as Frontiers. It's great to see so many of the audience coming all over the world, especially to listen to this project that I'm the coordinator of, NANOSPRESSO, and I'm going to explain the technical details of this concept. Because what we are trying to do is actually take nucleic acids, which are a marvelous class of therapeutics, but come with difficulties, combine them with nanoparticles and actually do that at the bedside. And I think that that is one of the enabling technologies that allows these particles also to be used in rare diseases, where it's difficult to make a business case, and it's really an important asset to democratize access to these types of advanced therapeutics. So let's start with the hype, with the enthusiasm about nucleic acid therapeutics first. So, well, this is the central dogma of molecular biology, the flow of information, where a gene is encoding a protein in the DNA, and it's transcribed into messenger RNA, and then ultimately it arrives at, or is translated into the workhorses in every cell, the proteins. And if there's something wrong in this flow of information, either the gene is mutated or the messenger RNA is wrongly processed, you can imagine that there are issues appearing in the workhorses, in the proteins, and that will cause disease. And correcting correcting this flow of information can be done with nucleic acids, and with that, it offers almost unlimited potential to treat diseases. So you can imagine that, for example, if there's a mutation in the DNA that is transcribed and ultimately in a mutated messenger RNA, you could either correct it in the DNA all the way on the left side, you could supplement the correct messenger RNA in the middle, or you could block this pathological messenger RNA by coupling something like an antisense to it, making it impossible to be translated into the protein. So with nucleic acids, you can interfere in this flow of information and allow new therapeutic interventions to take place. And this figure illustrates the variety of possibilities that exist with nucleic acids. It basically can do anything you could want and more from a therapeutic. You can make a therapeutic, you can make more of a therapeutic that is listed under 1 and 2, but you can also do the opposite, prevent under number 7, prevent a protein to be actually made. And even under number 8, you could even interfere with the DNA or do DNA editing to actually cure diseases. And all these potential points of intervention are really remarkable from a therapeutic perspective, because this cannot be done with conventional therapeutics. At the same time, it's almost like it's really difficult because they are almost meant not to be drugs. They contain a lot of negative charges and their molecular weight is high. They can easily be degraded. But at the same time, they have an intracellular site of activity. And it cannot cross a membrane, because these large molecules, compared to small molecular weight drugs like aspirin, for example, a lot of negative charge need to pass through the cellular membrane in order to reach the cell interior to do all these activities. But that is an oily, fatty layer. And you can imagine that a bulky, large molecule that has a lot of negative charge, that is very water soluble, has difficulty in transporting through this barrier. So that makes it a really difficult molecule to work with. At the same time, what I already said, it does everything you could ask for and more. It also even allows you to interrogate undruggable targets for which we, with current small molecular conventional drugs, are unable to find cures for. It can also be curative. Many of the current drugs just suppress symptoms, but if you could edit the DNA, you could actually cure a disease, which is something that we've not been able to do, except for infectious diseases in the past. Also, we know how these compounds should work. A messenger RNA should translate into a protein. So your lead compound and the identification is very fast, and your mechanism of action testing is complete in weeks. And importantly, and this is really crucial when you want to develop a pharmaceutical formulation, essentially it's always the same molecule. It only differs in length and sequence. And that is something that is shown here. So on the right-hand side, you see proteins, but it also applies to conventional therapeutics. Whenever you start on a project, you work with your therapeutically active molecule, but you never know what type of characteristics it will possess. Will it be positively charged, neutrally charged, negatively charged? Will it be very sensitive to light, to humid conditions? You never know. So every project starts essentially from scratch. If you take the opposite, on the left-hand side, the nucleic acids, it's always essentially the same molecule. It only differs in length and sequence. By changing the letters in the alphabet, you change, for example, the messenger RNA and change the protein that will come out of it. But essentially, it is the same molecule. So the lessons that you learn while developing your formulation for the first time, they can be applied to the subsequent formulations. And that is really an asset in drug development. It can really speed up things. So how to overcome these difficulties? It is a wonderful molecule, but it is difficult to deliver. And there goes a big shout out to Professor Peter Cullis, University of British Columbia. He was one of the first to envision how this could be done and how this could be done with lipid nanoparticles. So with lipids, you can actually encapsulate the therapeutic nucleic acid into a nanoparticle. And that can be done by self-assembly because these lipids will organize themselves in an aqueous environment in such a way that they encapsulate the nucleic acids into nanoparticles of approximately 100 nanometers. Of course, this needs to be very carefully controlled. Otherwise, you get particles of all different sizes and they go all different places. But if you mastered the conditions under which these particles are manufactured, you get very reproducible, very nice nanoparticles. And these can help you deliver your therapeutic nucleic acid to the right site. So this is an advanced therapeutic which almost does everything you could want for. So it's intravenously injected. Subsequently, it arrives at the surface of the target cell. It is then internalized. The therapeutic nucleic acid is allowed to escape by the help of the lipid nanoparticle, and then the therapeutic nucleic acid can do its job, in this case, nuclear translocation, integration, and payload expression. But essentially, all of the therapeutic nucleic acids, once they have entered the cytoplasm, they could do their therapeutic activity. And the first lipid nanoparticle filled with a therapeutic nucleic acid came on the market in 2018. That was Onpattro. Not a moment too soon, you could argue, because that allowed us to build upon that knowledge that was generated during the production of that particular drug molecule during the COVID pandemic, of course. And that is not a moment too soon, because within a year we were confronted with a pandemic that required us to really learn from all the lessons that we've learned on the left-hand side from patisiran. So this drug, patisiran, provided a recipe of how to make lipid nanoparticles loaded with a therapeutic nucleic acid. You can see a variety of different payloads. So you see the siRNA for a particular type of amyloidosis on the left-hand side in patisiran. Then you have the two COVID vaccines in the middle and a new CRISPR-Cas-based advanced formulation that is currently in clinical trials on the right-hand side. And essentially, they are all built on that same recipe of 4 lipids together with the nucleic acid, form a nanoparticle and can be used therapeutically. If you look at the COVID vaccines, of course, everybody was focused on scale-up. During COVID, new factories emerged where lipid nanoparticles were churned out by the kilogram. At the same time, in our lab, we make lipid nanoparticles, and I think I can say we can make nanoparticles at a very high quality. I would gladly inject myself with these lipid nanoparticles because I think they match the quality of industrially produced lipid nanoparticles. And that can be done by the virtue of microfluidic mixing. You can get very reproducible mixing if you do this on a microscopic scale. So the self-assembly can occur in these types of microfluidic chips. Here you see the 2 fluid streams, one yellow, one red, resulting in an orange mixture. And that is essentially what needs to be done. The nucleic acid needs to be encapsulated in the lipid nanoparticle under reproducible conditions. And within such a microfluidic chip, you can essentially always replicate the same conditions, provided that you keep the flow rate and the flow rate ratio constant. And that, well, the fact that we produced in our lab high quality nanomedicines, which I gladly inject into myself, was basically the foundation for NANOSPRESSO. Not in the least because I worked together with Huub Schellekens at one point, who devised or thought about the Biospresso. That was perhaps a bit too early for its time. He envisioned a individualized protein therapeutic production facility on your kitchen top where bacteria would be engineered to produce the antibody of choice that would help you cure your therapy. That is still difficult, I think, but the thought lingered in my mind. And I think with nucleic acids, all of a sudden, the concept can become a reality, because combining nucleic acids and lipids in a blister and do microfluidic mixing inside a cartridge should allow you to give always reproducible lipid nanoparticles. Combine that with a cartridge inside an activation station, and that activation station should take away all the burden from the hospital pharmacist regarding the quality control. So provided that the pressure buildup in your cartridge and all the loading controls and volume dispenses has happened correctly, the green light should appear, and as a result, the hospital pharmacist should sign off on the quality, because all of the quality controls is embedded within the activation station and doesn't require subsequent testing of the final product. And then you arrive at a vision like this, where on the left-hand side, you obviously have your Nespresso machine with all the tastes and that are available. And on the right-hand side, you have your activation status with the different cartridges filled with the nucleic acid of choice that is therapeutically active for an individual patient. You load the cartridge, put them in the activation station, and as a result, it will produce the high-quality lipid nanoparticles within your hospital pharmacy. There are new developments going on. This is an example of CureVac's messenger RNA printer, as they call it, where in a completely automated fashion, even locally within a hospital, you could produce GMP-grade messenger RNA. And also companies like Abogen have a completely automated suite in which they can make messenger RNAs on demand. So you also see there that from a production perspective of the active ingredient, that even is scaling down into local hospital settings. So with that, on the left-hand side, we have our ingredients supplied with a variety of companies that participate within this consortium. And within the middle, we have our academic institutes that participate to try and make NANOSPRESSO a reality, where we have the machine, the characterization and performance, and of course, all the legal regulation and societal challenges that surround it. And finally, of course, we all do this to benefit the patients. With that, thanks a lot, and I hand over to Lee.
Thanks a lot, Ray, for that beautiful introduction to the NANOSPRESSO concept. It sets a really good basis for today's discussions. Next presenter is Sabine Fuchs, Professor of Metabolic Diseases and Innovative Therapies at the University Medical Center Utrecht in, of course, the Netherlands. Sabine's a principal investigator at the Regenerative Medicine Center Utrecht at the Heumber Institute and founder of the award-winning company Hello R&D. She uses innovative technologies such as liver organoids and gene editing to develop treatments to address serious, significant unmet needs among patients with metabolic diseases. She also mentors clinicians and scientists through programs such as Tulips, the training upcoming leaders in pediatric sciences, and the Eureka Certificate Program for Translational Medicine. So welcome, Sabine, over to you.
Thank you very much, Lee, and thank you for this opportunity to present here. I will discuss the clinical aspects of the NANOSPRESSO project. And I am a metabolic pediatrician, and I think to get us all on the same page, I'll explain briefly what a metabolic disease is. So a metabolic disease is a genetic disease where a mutation causes a specific enzyme to dysfunction or not function sufficiently. And normally in metabolic diseases, this enzyme is involved in a metabolic process. And usually this entails the conversion of a substance A into a substance B. And when this does not happen in the right way, then substance A will not be converted into substance B, and it can lead to accumulation of a toxic compound and thereby cause symptoms, or it can lead to a deficiency of substance B, a compound that might be needed for the functioning of the organism. And depending on the enzyme or the specific mutation, essentially any organ can be affected and any symptom can be a presenting symptom occurring at any age. In the Netherlands, because that's where I work, we know that approximately 10,000 families are affected by one child or more children with a metabolic disease. And with the integration of more and more genetic screening, this number is expanding gradually. Many of these diseases are untreatable, but for some diseases we do have treatments. And we've focused with our treatments historically on the metabolites that are altered. So if one of the substances become toxic, then we try to avoid production of this substance by giving patients, treating them with a diet. On the other hand, the problems may also arise because of the deficiency of a certain substance, and we've been used to treat that by providing that substance. But this can only treat a subset of patients. With evolving possibilities, it became possible to also directly target the enzyme that dysfunctions, and we can do that either by providing a cofactor for the enzyme, or we can deliver the enzyme in a pure form by itself, or through an organ that produces the enzyme, which in the case of metabolic diseases is often the liver, because the liver is a very important metabolic organ. But it might also, for example, be hematopoietic stem cells, because they can also cross the blood-brain barrier. But even with these therapies, still many diseases stay without a good treatment. And I've been thinking, why are we so poor or so bad still at treating these diseases? And I think it is because metabolic diseases represent such a large group of so many different individually very rare diseases. And we've always been focused to try and really understand one of those diseases and try and develop a treatment for that. But with the increasing number of diseases, which are now over, well, for metabolic disease, we know now that there are over 1,900 diseases, but for rare orphan diseases, even over 8,000 diseases. So I think it is now time to try and look for a more general approach, which we can then target to the individual rare disease. And when I think about what is general among all these diseases, is that they are all genetic diseases that result in an enzyme deficiency. So when focusing on that genetic cause of disease, I will first focus on the affected mRNA that Ray also talked about already. So mRNA can be used now as a therapeutic, and it has been used in the COVID-19 vaccine, of course. And Ray already mentioned Peter Cullis as a key person in developing mRNA medicines, but Katalin Kariko is certainly also a key person in trying to make these mRNAs usable for medical application. Once you use this technology, the mRNA encapsulated in lipid nanoparticles, but inject them intravenously, then almost all the lipid nanoparticles accumulate in the liver. And these are experiments that were actually done in Ray's lab, where you can see that we used a reporter mouse in which the mouse was administered with an mRNA that turns the cells red. And here you can see the organs in which most of the cells turn red, and you can see that with increasing dosages, almost all liver cells are reached with this lipid nanoparticle. So we thought that is really interesting for metabolic diseases, because the liver is such an important organ for metabolic diseases. Maybe we can give the deficient mRNA or the mRNA of the deficient enzyme to treat patients with metabolic diseases. Unfortunately, Moderna thought the same, and they developed a treatment, a first-in-human trial for two different metabolic diseases, propionic acidemia and methylmalonic acidemia. And these are two severe metabolic diseases in which the liver cannot metabolize certain amino acids, and that results in the production of toxic compounds that can affect different organs, like, for example, the brain and the kidneys, but also the heart and also the optical nerve, so patients can become blind. So it's a severe disease for which currently we don't have a good enough treatment. The treatment that we are testing right now is the mRNA treatment for MMA. That's what I'm talking about, but we also host the PA trial. But for MMA, we encapsulate the encoding mRNA that encodes the deficient enzyme, which is the MUT enzyme in this case. It's injected intravenously in the liver. The formulation will arrive and the mRNA will enter the cells where it can be translated into the MUT enzyme, which can then be translocated in the normal physiological way to the mitochondria, where it will dimerize into the homodimer, which can then perform its activity. This is an international study, which is a dose escalation study. And here you can see the first results of the first 3 cohorts, which were the lower dosing cohorts. And we are now in cohort 6, in which patients receive higher doses of 1.2 milligrams per kilogram already. But what you can see is that we here look at the toxic products that are produced in the patients. It's methylmalonic acid and 2-methyl citrate. And you can see that in the first cohort where we used a very low dose, there was no effect. But with increasing dosages, you can see that there is an increasing effect in reduction of the production of the toxic compounds. And with the higher cohorts, we even see this stronger. We have treated 3 patients in the Netherlands now. This is one of the patients. He started in cohort 3, and he started in cohort 4 actually. And he was a very shy boy with not very much energy. And upon treatment with this treatment, we can see that he became much more energetic. And the parents really say it's a totally different child. And actually, we see remarkable clinical effects in all the patients that have been treated so far. For the propionic acidemia trial, the interim analyses have already been published, and there they also see beneficial effects of the treatment. And by now, there are mRNA trials that are either running or being prepared for a number of other metabolic diseases, which primarily affect the liver. The major limitation, however, is that the mRNA expression is only transient. So it is only expressed for a couple of days, and then the enzyme is expressed for a couple of days, but then you see that the effects are fading. And actually in our older patients, they really report that they feel well after the treatment, but after a week, they start already feeling symptoms again. And that means that these patients really need to come to the hospital every 2 weeks to receive an intravenous injection for their medication. So I think this is a huge development for metabolic diseases, but we're not there yet. And I think we can now focus really on the disease-causing mutation. And I think that is what we should do. Because this was enabled by the CRISPR-Cas9 technology, which was first described by Jennifer Doudna and Emmanuel Charpentier, who won the Nobel Prize in Chemistry in 2020. And they showed that the CRISPR-Cas system can be used as a very precise molecular scissor that can cut the DNA at an exact location wherever you want to cut it, basically. This is really exciting, and you can really cut the DNA at a place. You can use it to repair a genetic mutation, but that is quite an inefficient process. And rather, the cell will react on the double strand cut in the DNA by trying to repair it directly, which is a sloppy procedure and which leads to the integration of insertions, or can insert deletions in the DNA, which will rather silence the gene than correct it. So that is why David Liu thought of using a slightly less active Cas9 enzyme, a nicking Cas9, and if you fuse that to another enzyme, for example, a deaminase enzyme, you can deaminase certain bases and thereby convert, for example, an A into a G or a C into a T nucleoside. So this is very specific and really can help to correct a mutation, but it's only applicable to very specific mutations. So then David Liu thought of another system, and he thought of combining and fusing the nicking Cas9 enzyme to a reverse transcriptase enzyme, which is basically a writing enzyme that can rewrite parts of the DNA up to 20 nucleotides, but by now even longer stretches. And thereby you can essentially edit any mutation that you want to edit. So what if we would exploit this transient expression for direct in vivo gene editing? So giving the modified RNA that encodes the gene editing tools, encapsulate that in lipid nanoparticles, inject that intravenously, and then it will arrive in the liver where it will be expressed. It can then correct the mutation. And because the expression is only transient, the tools will be cleared and you will have a permanent cure of the cells with this transient treatment. So we think this is really a game changer. And I think that because for a lot of metabolic diseases, we know that we only need to correct approximately 5% of liver cells to resolve most of the symptoms of the patients. And in the first animal studies that were done using these lipid nanoparticles, we saw that for base editing for a certain disease, we could reach much higher levels in liver cells of mice and also in non-human primates. And we also saw that it was safe. And by now, the first human trials are ongoing for 2 different liver-dominant diseases, and in both beneficial results have been reported already, but these are, of course, preliminary yet. To target other organs, another system has been developed in which you can encapsulate the ribonucleoproteins in virus-like particles. And the very first animal studies for that have also shown quite promising results, I'd say, in mice, where they've seen that over 60% of liver cells were edited after 1 intravenous injection, and also retinal cells and even brain cells could be targeted after localized injections. So we tried to use this to develop a gene correction therapy program where we have developed a specific reporter that we can use to tailor our gene editing tools to the patient-specific mutation and the mutational context. We have a large biobank with organoids from a lot of patients and from different organs that we then use to target and further refine our treatment to the organ-specific and patient-specific context. And then we aim to develop these into personalized therapies using the NANOSPRESSO approach. And we first focused mainly on the liver because it's the easiest organ to target, but with some recent financial support, we are now also focusing on other relatively easy to target organs like the bone marrow cells, the eye, and not easy to target, but very, very wanted for metabolic diseases, the brain. And this may sound like science fiction, but just a month ago, it has been shown that this is really possible. This is in the United States, where they've treated a very first patient who was diagnosed after birth with a rare genetic metabolic disease. And within 6 months, the treating team, in a collaboration with regulatory authorities, but also with industrial partners, they were able to transform this lethal diagnosis into a treatable condition. And they've treated this patient now with 3 doses, I think, of base editing treatment to try and correct the underlying genetic disease. Of course, this was the ideal candidate, the ideal patient for this situation. This was a patient with a liver-dominant metabolic disease in which we know that the liver is the easiest organ to target, and this is a disease where you don't need to target all the cells to resolve the symptoms. Parents were open to this. This was a base editable mutation. That's also very important because base editing is still more efficient and more precise than prime editing. So I think if we want to translate this treatment strategy to the many different patients with metabolic, but also other genetic diseases, we will need to also to try and target also other organs, but also involve prime editing, because with prime editing, you can address essentially any mutation. And also for prime editing, there are encouraging developments to try and develop these also into clinical therapies. So with that, I think it is prime time for patients with metabolic diseases, because in addition to the treatments that I explained already before, we now have new possibilities using mRNA therapies, but also the precise gene correction therapies, in addition to the gene therapies, the gene augmentation therapies that I didn't mention today. It's not only us, the researchers and the medical doctors that are excited about this, but also the patients themselves who try to gather money, to collect money, to invest in these developments. And I think we can really make this happen if we employ strategies like NANOSPRESSO, and we think about these therapies as a platform technology that we can adapt and quickly adapt to the different diseases, and thereby develop therapies for the many patients for whom we don't have good therapies yet.
Wonderful. Thank you so much, Sabine. What a beautiful presentation and an ending on a note of hope there. Our final speaker is Olivia Lewis. Olivia is a PhD student specializing in drug regulatory science at Utrecht University. As part of the NANOSPRESSO project, her research is investigating regulatory pathways and challenges related to localized preparation of high-quality, personalized nucleic acid medicines, as we've been hearing about today. She's motivated by the complex transdisciplinary issues that bridge science and society, aiming to improve public health outcomes through these interventions. So, Olivia, thanks for being here with us, and over to you.
Thank you. So yeah, as mentioned, I'm working on the regulatory challenges for NANOSPRESSO, which, as you can imagine, are quite complex. So with that in mind, I don't tackle these things on my own. I work as part of a much broader team. We're very interdisciplinary. We tackle the regulatory challenges, health technology assessment, legal frameworks, and also the operational feasibility. Yeah, I just wanted to give my team a shout out because they're very capable and very fun to work with. So with that, the regulatory challenges in NANOSPRESSO can be divided into three main categories. We have the fact that we're using nucleic acid therapies, the lipid nanoparticle delivery system, and also we're trying to do decentralized small-scale production methods. And each of these things have their own challenges. So to tackle this, we have quite a lot of ongoing research topics in all of these areas being tackled by, the extended team. The three topics on the left-hand side are kind of where my research lies. So I look at more the centralized kind of environment and why it is so difficult for us to go on a centralized route for RNA regulation purposes, lipid nanoparticle regulatory purposes, and also what the proposed EU legislative change means for us. And on the left-hand side, the broader team is really doing a lot to kind of look at pharmaceutical compounding and the readiness in Europe for doing something like that. So firstly, I'm going to talk about the regulatory challenges for nucleic acid therapies. As Ray has already covered, they have vast potential. So they work at different molecular levels, different mechanisms of action, and they can be more transient or more permanent in their effects. So overall, it's quite a complicated and diverse group. So I'm just going to take one example here, which is Melisen. I'm sure many of you have heard of this one before. So this was the first personalized antisense oligonucleotide treatment designed for a girl called Melisen after her, makes sense. But is this an advanced therapy in the European regulatory system? So really to answer this question, how does RNA actually look in the current regulatory system? Well, there are two key decision points, and it's whether they target an infectious disease. If they do, they're on an entirely separate regulatory pathway where they are categorized as an immunological medicinal product. And for the purpose of this presentation, I'm not talking about them. So that leaves the production method. Now, only mRNA really is still biologically produced. Even that is evolving with technological advances. and the rest are chemically synthesized. And the reason that this matters is because, advanced therapies must be biologically produced as stipulated within the European regulations. So this means that the rest of the RNAs, so things like antisense oligonucleotides, everything that is chemically produced, is classed as a chemical medicinal product. Okay. So this means that Melisen is actually not an advanced therapy. And this has downstream implications. So it means it can't qualify for things like hospital exemption routes, like personalized gene therapies could. But also the legislative changes do propose to amend this ATMP definition to include synthetic medicines as well. But I just want to kind of draw attention to the fact that the situation is a little bit more complicated than that. Okay, so they don't fit neatly into the existing categories. They are more complex than chemical medicinal products, but they don't quite meet the criteria for advanced therapeutics, and that is because of the transient effects as well as the synthesis. And this is known by the European Medicines Agency as well, that have highlighted the lack of consensus on the definition of RNA-based medicinal products. And the reason that this really matters is because developers really do need the tools and resources to know the regulatory pathways available. This kind of informs decision-making, the clinical trials that we want to conduct, and evidence requirements. So this also affects accessibility. In an ideal world, we would put this through a centralized pathway. It could be reimbursable and centralized, but it's more complicated than that. So I did a study to kind of look at, how we could categorize and define RNA from a regulatory perspective. For this, I looked at all of the European market authorized RNA products that are currently out there. I really wanted to look at their characteristics and see if they had commonalities that had regulatory implications. The reason I wanted to do this was to kind of see if there was a regulatory basis for grouping them. And there were 13 products overall. 7 were ASOs, 5 small interfering RNAs, and 1 aptamer. They almost all targeted the liver. They almost all targeted rare diseases. But despite this homogeneity, there were some commonalities in the subtypes of RNA and the risk profile that regulators were really paying attention to. So an example of this is the RNase H-mediated ASO, which often has a very particular chemical modification called the 2-MOE modification. And every time this has this modification, we were seeing in the regulatory dossiers that there was an affiliated risk of coagulation abnormalities. And that's regardless of the sequence. So based on this, we are seeing kind of trends within the products that are irrespective of the sequence. And this is also important to mention that because a lot of them target rare diseases, they had very similar missing information or uncertainties. And to me, all of this points towards, the need to kind of form a basis or a categorization system whereby we can refer to, this definition to kind of lower that threshold for approval and kind of build on existing knowledge, leverage that knowledge for new products. So with this in mind, I kind of developed this, categorization system, which is what regulators are already kind of doing. So they are already making these considerations, but I've just put it into a decision tree, essentially. So we're still going to be considering the chemical synthesis or the biological production, as that does have an impact in terms of the regulation. But in terms of for RNAs, they do have characteristics that have regulatory implications. So you can have binding, sequence-specific hybridization, or insertion of an entirely new sequence. And these are the 3 kind of main broad categories. And then you can have subdivisions for the common chemistries that do impact the safety profile. And the benefit of this is that it will enable the pooling of data and then permit us to use prior knowledge, which is essential against rare diseases with small patient populations where you are restricted in your ability to gather kind of mass scale clinical trial data. And especially we're talking about something like NANOSPRESSO, this kind of thing matters because we envision it as a platform technology. To very, very briefly discuss lipid nanoparticles, it's only 2 slides, I promise. So in this device, in NANOSPRESSO, the lipid nanoparticle is not the active substance, and that is within the European regulatory system overall. In the COVID vaccines, it's considered an excipient, not an active substance. It is always just a carrier, an excipient. The nucleic acid that it carries is considered to be the active pharmaceutical ingredient. So how do regulators look at the lipid nanoparticle component? Well, the reason I'm interested in looking into this is because the lipid nanoparticle does have immunogenicity effects, and we know this especially after COVID, but there is no definitive framework for regulating them. So I really wanted to kind of look into this and see if there's any characteristics that do impact the safety profile for lipid nanoparticle containing products. And this will really help us to advise the developers, in our case, NANOSPRESSO, for what kind of evidence they might need in terms of controlling the safety aspects of a lipid nanoparticle in a platform technology. And importantly, the findings that we're going to gather from all of these areas should have impact for the broader community and not just for NANOSPRESSO. This research is to be continued. It's only in the question stages right now. Thank you.
Thank you. Thank you very much, Olivia. I'm sure we'll return to the complexities of regulation probably in the discussion section to follow. So thank you very much. It's time now to move to the discussion section, and please keep your questions coming in. We have many already, and we're going to try and get through as many as we can. Before we do so, I'm delighted to welcome 2 more guests. First of all, Dr. Alexandre Ceccaldi is General Secretary at the European Technology Platform on Nanomedicine, the driving force behind Europe's nanomedicine agenda. This is a network of over 130 academic, industrial, and healthcare organizations. Alex also represents ATPN on the Strategic Advisory Board of ERA for Health at the partnership, and also the Nobel European Project as well. And also, I'm very pleased to welcome Alexandra Heumber, the CEO of Rare Diseases International. Alexandra is an accomplished global health leader, dedicated her entire career to improving healthcare policies to people living with a variety of diseases, including through the International Alliance for Myalgic Encephalitis and head of policy for the Neglected Diseases Initiative. In a current role of Rare Diseases International, she drives global strategies to raise awareness about rare diseases and improve access to diagnostics and treatments, and advocating for the rights of patients with rare diseases worldwide. So welcome to you both as well for joining us today. And perhaps, Alexandra, if I could start by asking you, we've heard today mainly a focus on rare disease patients. The article, I should say, the lead article on NANOSPRESSO points to the importance of mRNA therapies and other kinds of therapies in a variety of diseases, but the main focus at the moment is on rare diseases. So from your perspective, leading Rare Diseases International, what unmet need, what's the status of the unmet need, and what are your first reflections on how a decentralized approach to providing advanced therapies might fulfill the needs, the unmet needs that exist in the world today? Please.
Thank you very much, Lee. Thank you, Frontier Science, to invite me representing Rare Diseases International. So Rare Diseases International is the global alliance of persons living with rare disease across the world. So indeed, I think it's important to first underscore the scale of rare diseases as a public health issue. Globally, there is over 300 million people living with a rare disease, yet 95% of rare diseases still have no approved treatment. That means millions of families worldwide facing not only the health impact, but also enormous social and economic burden, often without hope of an effective therapy. So the challenge is that traditional pharmaceutical models are not designed to address the small, dispersed patient population. And higher research and development cost, low expected return most of the time, regulatory complexity, as has been just shown, and limited reimbursement in many countries, all of that contribute to a huge unmet need. In short, there is a profound structural gap in treatment access for rare diseases globally. And closing the gap is fundamentally an equity issue. So while ADI doesn't have the competence to assess the scientific aspect of models such as NANOSPRESSO, we see a great potential in innovative process to improve access and equity. And I would highlight the four main points. The first, in our policy outlook, we refer to equity through innovation. And this is really important to us at ADI. It means that technology innovation isn't an end in itself. It has to be designed from the very beginning, and it has to be deployed to close the access gap, not widen them. So platforms like NANOSPRESSO can help make highly advanced therapies available in settings that would otherwise be excluded by reducing the dependency on centralized high-cost manufacturing. The second aspect is that, as you know, rare diseases often involve ultra-small patient group spread across many regions, so making the traditional centralized production costly and unpractical. So localized and magistral production model, such as NANOSPRESSO, could deliver truly personalized nucleic acid therapies closer to patients, so faster and potentially at a lower cost. And then also it creates opportunity for local capacity building, skill development, and ownership, so that innovation benefit the health system overall, and not just the company that bring the innovation. And then lastly, by making production more flexible and scalable, such model can also enable equitable distribution of treatment, even for ultra-rare conditions. So in short, they can embody the idea that innovation really have to serve everyone, everywhere, not just patients in wealthy, highly resourced settings.
Super. Thank you very much, Alexandra. That's a very full answer. And I do, everybody online, if you go to the Frontiers in Science lead article, the hub page, you can see the link there to the policy outlook article that Alexandra and her colleague provided for the hub, and where we elaborate on those points. We've had quite a lot of questions looping back round to the NANOSPRESSO and the technologies involved. And Ray, if I could come first of all to you to dig in a little bit about those. A few questions, I'm going to group them together so we get through as many as we can, on things like standardization and ensuring safety, and sort of quality control aspects, if you like. So could you just outline what are the advances in the lipid nanoparticle technologies, the microfluidics, and the processes and the new ways of not only providing these, but also monitoring the outputs that are allowing NANOSPRESSO to provide a safe, effective product locally in this way?
Yeah, I think that over the years we've learned now a lot about these lipid nanoparticles, how they assemble, and what the conditions are to make them work. And as I said, I was really struck by the moment that we made to incubate with cells a batch of high quality lipid nanoparticles, and I thought, well, this would be on par with what industry would produce. It's as good. I know for sure that the particle size is okay, that all the nucleic acids are inside, and that it conforms to any test that you could do. So that was really a moment where I thought, well, this is great. We can make it on a small scale, but can we even automate it so that you don't have to know it all to still be able to use it, and transfer the quality control from batch to batch, taking samples and then testing them and saying, well, it conforms to all these 25 assays and I've consumed half of the product while doing this, to an onboard quality control, because we know what needs to be measured. And the moment that we mix nucleic acids with lipids, this self-assembly process starts. And one of the crucial aspects is that the moment that this does not happen in the correct fashion, one of the earliest things that goes wrong is the particle size. So either it will not be able to pass through our final 0.2 micrometer filter because it became too large, or it will not have this typical opalescent color, so to speak, of the final vial. So just with these two quality controls, you're already halfway there, I would say, that this machine operates. With the rest, we have all full onboard control and logging of all the intermediate steps. So does the pressure to the blisters build up correctly? Are there no leaks? Are the volumes that are dispensed of the correct size? And that will guarantee that the outcome will always be the same. So those are going from quality control of batches towards quality control of the process inside the machine. That is where we want to arrive at.
Right, thank you very much. Sabine, a few questions on clinical applications. You outlined some of the clinical studies for some of the therapies, so the nucleic acid therapies. With respect to the NANOSPRESSO device and these therapies, what's the next step in the clinical development, the applications? And we had a particular question from David van Bennekom from the European Rare Kidney Disease European Reference Network, who asks, would it be possible to target the kidneys, and are there particular challenges in this?
So thank you for this question. Currently, we're mainly focusing on the liver, and I think that is the organ that we can reach because the lipid nanoparticles naturally accumulate in the liver. So liver-dominant rare diseases where we know that we don't have to reach all the cells to treat the patients are the diseases that we focus on currently. The project focused on providing the mRNA of the deficient enzyme, but I think with improving CRISPR-Cas technologies and also having the first example of that this is really working in actual patients, I think the next step will be to develop personalized gene correction therapies, as I tried to explain in my presentation. As for the other organs, we can already target some of the immunological cells, so bone marrow-derived cells, so blood cells, like for sickle cell anemia or thalassemia. And next steps will be trying to target other organs like, for example, the kidney. The kidney is, well, at this very moment, not possible to reach with enough efficiency using lipid nanoparticles because there's just too much that accumulates in the liver. But I think that in the future, with active targeting of lipid nanoparticles or use of other vehicles like the virus-like particles or viruses, this will be a possibility.
Wonderful. Thank you very much, Sabine, for that answer. Incidentally,
and maybe I can add, now that I'm thinking of it, of course, we can treat nephrologic or kidney diseases that are caused by the accumulation of toxic products that are generated in the liver. So, for example, in methylmalonic acidemia or hyperoxaluria, these are kidney diseases that can be treated by treating the liver.
Absolutely, well taken. Thank you. Just in case, incidentally, if my signal goes down, there's a slight technical problem before. Don't be alarmed. My colleagues will step in. Everybody. But so far so good. Olivia, some questions with respect to thinking about regulation. What have we learned so far on the challenges of centralized production of these medicines, and with respect to magistral or local compounding, despite the advantages, it comes, as you sort of alluded to, with a specific regulatory nuances and challenges. And so where does, you kind of left on that jumping off point there, could we just flesh out a little bit, and Ray, maybe dive in as well, where do we stand with the regulatory situation of magistral local production as opposed to centralized production and therefore sort of a centralized approach to regulation as well?
Wow, very long-winded question.
Thank you.
That's regulation, I suppose. So I would say the centralized system is just really, it's not built with something like NANOSPRESSO in mind, like at all. We have multiple components that make it even more complicated. So we have, as I kind of addressed, the RNA within itself, it has challenges in the centralized system. Lipid nanoparticle, which isn't even an active product, it's an excipient, but with effects. And then, obviously the decentralized production is technically considered by regulators to be a loophole. But in this kind of instance, we really want to take advantage of that because the centralized system is just not built for us. And in general, it's not really built for, medicines that are targeting like ultra rare diseases, because you shouldn't have to go through these regulatory hurdles and all the costs of kind of getting a brand new drug centrally authorized. Every single time you have a very slightly different, sequence. So that's why we're doing some of the studies that we're doing and the things that I presented about, kind of grouping RNAs and whatnot, is because we do envision it to be more of a platform technology where we can kind of build on existing knowledge, switch out certain sequences, but not have to go through the laborious kind of process of having an entirely new drug every time for every different rare disease. And magistral compounding obviously comes with its downsides as well. But for something like NANOSPRESSO, we think it will be good for timeliness, for cost, and overall just reduce barriers to access. But importantly, it's quite a complex situation, which my colleagues are looking into, as it varies between different countries. They have different like national legislations that we really need to delve into and see the how we can facilitate that internationally.
Ray, did you want to add something there? I saw you nodding.
Yeah, no, definitely. I think that is the difficulty of it also. Indeed, even for compounding, you have to refer to text in the European Pharmacopoeia, so that also already requires a lot of sort of procedural thinking. But that is happening. So also within the previous consortium that I coordinated, Expert, we've tried to contribute to monographs on messenger RNAs that go into lipid nanoparticles that carry messenger RNA for infectious diseases as vaccines. So there's a movement to try and make this possible. And indeed, I think we need to enable or maximize the ability of individual pharmacists to be able to make these types of complex nanoparticles. And I think that the field would be sort of bit anxious, perhaps, these practitioners to touch this subject, but by making it as easy for them as possible, we could get to these individualized treatments. And to me, I'm very naive, so I'm trying to maximize this loophole as much as we can, because the moment that a doctor like Sabine prescribes an individual medicine for an ultra rare disease patient, the hospital pharmacist makes it and confirms that it has all the qualities necessary to be administered. It never sees the market. So this allows you to have a large acceleration of development, in my opinion. But I'm the optimistic technical guy in the lab.
Just to follow up on that, we've had a question from one of our viewers in China, actually, and the question was with respect to, in a decentralized model like NANOSPRESSO, how is liability managed in the case of adverse events? Does the liability fall on the platform provider, the technology provider, the hospital pharmacist, or the prescriber?
Good question. No, that is something that really is important to because as it works now, so there's the prescribe. As it works now, if we compare it to regular compounding, then the liability lies in the hand of the manufacturer of the medicine. So the hospital pharmacist is in the end the person who signs off on the quality of the formulation to be administered to the patient. So that is where I think also with the NANOSPRESSO machine, we have to really ensure that the machine gives them the confidence to sign off on this, and that the machine is always right regarding the green light saying this formulation is safe to administer. And actually, then the liability actually is very much aligned. So there is this direct contact between the formulation and the patient. So there's this one-on-one link between the product and the patient, and that is rarely so for any centralized administration. So there actually this link is much looser, and it is in some cases very difficult to find out whether something is wrong with a medication because of these loose connections where it takes many patients in order to find out that there's something wrong. So,
right. Alex, if I could come to you, Alex Ceccaldi from ETPN. I saw you nodding there. I guess you must come across these issues a lot in your role from a sort of network perspective. And before we get into that, from your perspective, to broaden things out, where does Europe stand in nanotechnology, medical technology development? How is Europe at the moment? And where does where does Europe need to focus efforts to lead in this context and make sure the continent benefits from the development? And what's your platform doing to promote that?
Well, if I may start by this, Lee, can you imagine how lucky I am to work with these people? We're talking about, no, I'm serious. I'm talking about, it's not that usual that you are in a project when you have the clinician that sees the patients every day. You have the nanomedicine professors that can really think about the concept. You have the engineers that do even the sealing, the level of sealing that you need in the cartridge. And you have people like Olivia working on regulatory. Normally, regulatory is like the burden of, oh, we need to comply with, no. They treat regulatory issues as a research question. So to me, this place is you might think we are dreamers, but at least we do it all together. So for us, for the ETPN to participate in NANOSPRESSO is a pure joy and a total motivation because it's patients driven. Now, 2 words on nanomedicine. Basically, we try to be that think tank representing this community of nanomedicine developers in Europe. So from academic lab to small SMEs, large industry, but also national association, and to put them in contact, notably with the European Commission for strategic advocacy, funding, etc., and also translation to the clinic. What is the situation? Well, we have a fantastic community in Europe of people working with nanotechnologies to treat patients in a novel way. We are really strong on the academic side, probably leaders or co-leaders in the world, like really, really strong. We are not that bad in translation to market. Many of the new drugs that came out of nanomedicine were at least born as a concept in Europe, but then most probably raised in the US. And that's a very common situation that we all know. So our role is not only to fund nanomedicine like in a techno-push manner, not at all. I mean, it's part of our role to find the right money for the right ideas. But then our role is to be sure that the people that has the best ideas find the right ecosystem in Europe to stay in Europe and develop their drugs, medical device in Europe. Maybe you know that I think we are second in patents in biotech in general in Europe. But we used to be number 1 or close to number 1. So we somehow lost a little bit the leadership, would it be in biotech or in nanomedicine? But I see the opportunity. The opportunity is that, as you know, the situation is the US is kind of special, and people will need connection. And at least in Europe, for the last 20 years, the ETPN has shaped this community of people working together. So yeah, let's say mixed feelings. But the scientific level in nanomedicine in Europe is absolutely fantastic for sure.
Okay, thank you very much. Again, broadening out, Alexandra, you in your policy outlook, you highlight the World Health Assembly resolution on rare diseases that was recently passed. Your organization had a key role in the inception of that. Could you give us a flavor of how important that resolution is, and how a platform such as NANOSPRESSO, personalized, localized production, is aligned with it, let's say, or could be a vehicle to help further the goals of that landmark resolution? Sorry, Alexandra, you're still muted.
Here we go. So we indeed at Rare Disease International, we see the World Health Assembly resolution that was adopted in May as truly historic. For the first time ever, rare diseases is now formally recognized as a global public health priority by the WHO member states. So the resolution calls notably, and this is important, for the development of a 10-year global action plan to improve diagnosis, access to treatment, and care integration, social protection, and health system capacity. So it really commits also countries to include people living with rare disease in their national health strategy instead of, developing in silo. Now there is really the recognition that integrating is the way forward. So now, so rare disease patients are no longer invisible or excluded. But an important point linked to what we're talking today is also encourages innovation. Innovation as really part of the solution, recognizing that new approaches are really essential to close the treatment gap. I think perhaps today, if there is really this opportunity that we have, is to bring innovation, making it equitable, and engaging patient voice. And when the three are really coming together, this is where we're going to really be able to address the challenge for persons living with rare disease that still exists. So it aligns with the WHO resolution and the global action plan that will have to be developed, because it really requires that we strengthen health system capacity to provide diagnosis treatment. So by having those platforms like NANOSPRESSO, some innovative way of delivering more adaptable to the context of rare diseases, we will definitely create some, saving for healthcare system, or we will come with innovative solution. So this global action plan will have to be developed in 2 years. We hope, so WHO will be in charge of developing. There will be official consultation. We still have no clarity exactly when and what, but we will monitor that. And it's an opportunity for all stakeholders actually to come with some innovative solution that can be scalable and tailored to also the different region of the world. We don't want one fits for all. So voilà, so I stop here for that question, but this is the moment, I would say, to really come with some innovative solution to inform the policy.
And you spoke about capacity building before. If this sort of model is to benefit lower resource settings as well, how can we ensure that sort of capacity building is done? Will that be part of the national action plans that the global plan will sort of drive, if you like, to ensure that there's capacity building of the right sort, and presumably looking forward toward this kind of solution to be there, and therefore the sort of capacity that's necessary?
Yes, absolutely. I think there is nowadays in public health really the recognition that first we need to have sustainable solution. So we are not anymore in a kind of dependency of main donors. And the second issue anyways, that we are facing a terrible donor finance crisis. So we have to find some solution, and those solution has to also come from the people in the different countries. So in RDI, for example, we really support or enable our members in all the different regions to engage in this process of informing what should be that global action plan, for example. We do not want a very global solution that dictate what should be in the different country. It has to come from the bottom, but bottom up. So and so capacity building will be essential, but definitely technology transfer as well, and partnership with the local partners. We see in low setting area like in Africa, there are a dynamic there that we need to be able to build from. So this was actually the highlight I wanted to share about what could be the challenges of those type of innovation to be really accessible in low middle income country. Again, it would be look at the financing barriers that often there is in those countries, the infrastructure gaps. Often there is problem of training, personnel training, quality control system, regulatory oversight, and supply chain. And then the liability accountability, actually, I was happy that it came just earlier, because this is something that for decentralized model needs to be also looked at. And if we address those barriers, infrastructure gap, financing, regulatory alignment, liability accountability, and we really tackle it with capacity building, technology transfer, partnership with local health authority, and inclusion of local stakeholder from the outset, we can definitely have the right ecosystem.
Wonderful. Thank you. Thank you, Alexandra. So last few questions, I think. So this is another question from China, and I'm going to throw this one out there, and Ray, it might be for you, Sabine, you might dive into, maybe even Alex, I'm not sure. Do you foresee integrating AI-driven nucleic acid sequence design with the NANOSPRESSO rapid manufacturing system to create a fully automated end-to-end workflow from diagnosis through to bedside formulation?
Oh, I see Alex has his hand raised. So please go ahead, Alex. Oh, you're muted.
It's AI that thinks that I'm clueless. No, seriously, I mean, I'll just give you a clue. Indeed, AI is everywhere, and it is in nanomedicine. Would it be in the design of the particle, the association of the payload, the targeting, when, how much, etc. This year, for the first time, we had a full sessions in our event conference dedicated to AI, and it's a joke because in 5 years, AI will be at the core of all sessions that we do, right? So indeed, it will come. In the NANOSPRESSO, let's say, we have so much to do already without AI. And maybe I'm wrong, but some of the issues that we are targeting are not so easy to deal with, and I don't see AI integration right now super useful. But what is sure is that at the end, if we take the best of this technology, which is being a platform, not reinventing the wheel every time for each new patient, but have a core platform, then how do you play with the payloads, with the lipids in a few years? Indeed, yes. But at this stage, maybe it's a bit too early. We have big problems to solve before that.
Sabine.
If I may add to that, for the gene editing approaches, there we are using AI. So for rapid personalization of the guide RNAs, and especially the prime editing guide RNAs, there I think AI will be indispensable to really quickly optimize and personalize the gene editing tools to the specific patient and the specific mutation.
Yeah, I agree. So indeed, that to develop the next generation of platforms, what we are assembling now, all the data that we are gathering is so useful to fill a database or fill the artificial intelligence with all the data that we've got to try and build on that and make the next generation even better. I think that is where the innovation lies. But it's, yeah, no doubt that it will play a very important role from diagnostics all the way up to treatment.
Alex, please go ahead. I think you wanted to add something there.
No, just because maybe some people aren't familiar with nanomedicine, but the main issue of nanomedicine is that it has unique features, but the product is complex. You have so many key parameters, and basically the experiment plan, if you want to try all hypotheses by doing small variation of the core, the payload, the type, it's impossible to fund such research. So to navigate complexity, indeed, AI will be absolutely crucial in the coming years. It's already crucial. But I was talking more about the NANOSPRESSO project by itself right now.
Could you just build on that? Ray, in particular, actually Ray and Sabine, and maybe Olivia as well. When we have decentralized production and administration, and therefore not the same sort of coordination and data collection and R&D that's done in a centralized industrial process, how do you conceive of how data will be shared to ensure safety reporting on the levels of patient numbers to maximize those to make sure you see safety signals? And of course, evaluating efficacy in a large population as possible by collating data. Does the project envision data sharing as a sort of standard so that you still retain that sort of data collation that you would get in a normal clinical trial program?
No, definitely. And I think that Moderna actually put up a nice model to try and build this for the entire community of RNA or nucleic acid therapeutics developers. They propose a model, it's not there yet, but they propose a model where regulatory authorities would see all data that come in and you see the data of your own product. And by that, you allow the regulators to aggregate the knowledge and also identify potential safety risks on an aggregated level for the whole platform technologies, while you are developing your NS1s for each and every individual patient. And that may be guided in the end by the regulators. And I think that is a fantastic idea because it is platform technology. Everybody's working on their little thing, but there are issues that are at stake which are at a higher level, and the higher level should be coordinated from the regulatory side of things. And I think that this sandbox model where you have your regulator seeing everything and you are still responsible for reporting, first of all, but also getting back the feedback on your own project, I think that could be an interesting way forward.
Anybody else on that one? Data collection, Sabine?
No, I agree very much with Ray, actually. And I think the NANOSPRESSO platform, by devising your own medicines locally, I think it does allow for very nice N-of-1 trials where you can formulate also the placebo control, and you can play with having the patient on and off medication, which is usually quite difficult for patients which are really rare diseases, and you cannot you don't have access to a large cohort of patients. This is the way forward, I think, the N-of-1 trials. And I think the NANOSPRESSO consortium does allow you to do that. And then you need to integrate still all the data that you derive from that. But I think that will help to assess true efficacy from placebo effects.
Another question in, what do you think about the extensibility of this program beyond metabolic diseases, and I guess rare diseases too, say to autoimmune diseases or cancer, where gene correction could also be useful? So going beyond it, what we spoke briefly about some other organs, Sabine, before, but looking, if we could say, look in 10 years' time or beyond, where could this go more broadly beyond the rare diseases space entirely?
I think it can go already beyond the rare diseases spectrum. And I think actually, Ray, you are working both on cancer and immunological diseases. So maybe it's best answered by you.
Well, one of the areas. So I think that indeed, nowadays there's a trend to make every cancer almost individualized, and I think that is where one of the key opportunities lies. So if you again envision a patient journey from diagnosis, cancer is diagnosed, and also sequenced with its individual epitopes, your personal collection of immunogenic epitopes where you could be vaccinated against. That is translated into a messenger RNA, your personal messenger RNA cocktail on site in your messenger RNA printer, and then formulated on site with a lipid nanoparticle in a similar fashion as the COVID-19 vaccines to try and stimulate your immune system to recognize the cancer cells. I think that is already, I think, being developed by BioNTech, for example, and CureVac, to try and build this pipeline of activities where individual diagnosis, individual cocktail of messenger RNAs is translated into an individualized product. And I think that would be best served by doing it all on site.
Yeah. Last couple of questions, and maybe a few quick ones I can throw out there. This is from Hanine Ali Bouchetti from the University of Birmingham. Do you envision NANOSPRESSO being adapted for other drug delivery systems, such as polymer-based nanoparticles?
I think we can do. The moment that there's a self-assembling system that requires the encapsulation of a compound and is driven by the physical interaction, essentially, of your polymer and the drug, I think there's nothing against it in combining it. I mean, we've made polymeric micelles in the past, which is essentially the same process. There's the polymeric micelles, there's the drug. They need to be mixed, and provided you do that in the right ratio, in the right time, you get polymeric micelles with a very defined quality. So it should be easily possible. Yeah.
And perhaps the last question, and it might be, I'm not sure who's best to take this. Given that the accepted potential cost savings versus centralized industrial production exist, but what exactly do you think those cost savings are going to be? I guess, and I'm going to elaborate a little on the question here, in the sense that, as it was pointed out in one of the Viewpoint articles that we published in Frontiers in Science alongside the lead article, when there are other costs potentially of setup costs, training costs, equipment costs, capacity, and so forth, there'll be an ultimate trade-off between different costs, right? Does NanoSPRESSO expect to do a kind of a reconciliation or some sort of costing to demonstrate the cost savings associated with the platform, either notionally or in a real world data collection process? Ray, maybe Olivia, who's best?
Yeah, well, to start up perhaps first, so there is a complete program dedicated to health technology assessment to try and sketch all the different scenarios of all the different therapeutic intervention, as Sabine was already saying. So messenger RNA therapy is at this moment in time at least going to be costly anyhow, no matter how you produce it, whether centralized or localized. The cost of goods of messenger RNA are substantial and probably surpassing anything else that you do in the entire process. So there's perhaps limited opportunity there for cost savings. But if you think about gene editing technologies where a single intervention could potentially be curative, then you get a whole different scenario. But it's an integral part of NANOSPRESSO, trying to understand what the current costs are, what the current costs are of each of the technologies that are around the corner, and how if we use the NANOSPRESSO system, how this would change. It's a super important part because it will ultimately dictate whether it's in the end doable or not. It says all about the feasibility of the entire strategy.
Okay, Alex Ceccaldi, please go ahead.
I'm going to say something very simple, is that I understand that we need to think about the cost, but we need to think about the medical value. I mean, the current system doesn't work for rare disease. Some personalized, super high-tech drugs that could cure some patients are not affordable if you need to do the normal pathway with the huge costs of clinical trials and the insurance and so I mean, it's not just about cost saving. It's about finding a model where we can afford to develop a drug for 14 patients. And to me, we're talking about value and not just cost. But cost indeed will be evaluated. Indeed, I mean,
yeah. Well, Ron, I'm going to ask each of you to wrap up, if you like, in your take home. If you had in front of you the stakeholder that you would like to talk to in your sector with respect to nanomedicine development, with respect to NANOSPRESSO, with respect to the future of rare disease care worldwide, could I have your closing thought, if you like? And Alex Ceccaldi, maybe we could start with you from the perspective of ETPN. What would you like to say, the closing words with respect to NANOSPRESSO and beyond, given your perspective?
Well, when you are a technology platform, normally you think about technology. If I may, when you do nanomedicine, you spend 90% on nano and 10% on medicine at best. In NANOSPRESSO, we spend half on nano, half on medicine. And this is what I like the most. There's a momentum in Europe. You have the new Biotech Act. You have the new strategy for life science with a huge investment with like a strategy of 10 billion a year, plus really tactical tools now, like the sandbox for regulatory, etc. So there's an opportunity to test, I will say, crazy ideas. It might sound like this, but new ideas that can really take nanomedicine for its best, the unique platform settings. So to me, I mean, I'm not saying that it will solve all the problem, but it's worth trying hard with serious people that you have around the table. So yeah, our role is to create an ecosystem that even the most innovative ideas like that doesn't change the product, but change a full process, is absolutely worth trying. And I hope that Europe will totally be the right place to grow NANOSPRESSO.
Sabine, if you would please, from your perspective as a clinician, as a researcher.
I'd say that I think that the NANOSPRESSO approach will revolutionize the future for patients with rare diseases. And I think the future is now. It is now that it is possible to do this, and we can really provide even a curative option for patients. So I think we have to go for it now. Time is ticking.
Beautiful. Olivia, please.
Yep. So I would say, as we've just heard, the science is already there. You figured it out. Diseases are curable. And we as regulators really need to kind of make sure that we're now thinking along in the same mindset. As medicines are becoming more complex, more bioscientific, we really need to make sure that we're thinking in the same way, collaborating with the developers and ensuring access to these amazing developments.
And Alexandra, from RDI's perspective, how does this fit and what's your message to policymakers?
Yeah, no, really thank you, because for me it was so knowledgeable to hear the different perspective. I come from public affairs. RDI is a really global advocacy organization, but without innovation and without science, we will not address the needs of persons living with rare diseases. So we need to work together. So to close, I would say that we can see that when equitable innovation policy momentum, because indeed there is a policy momentum at EU level, like Alex mentioned, but also at international level, as I mentioned. And when there is real patient engagement, then, and when all those three come together, this is where we can really achieve, I would call it the paradigm shift, because rare disease is into a process of shifting, but it's up to us to make it happen. So thank you really for that, all the work that is being done by researchers, health practitioners, along the side of the community of rare disease patients.
And lastly, Ray, you brought this project to us. Your leadership of this project. How would you close the show?
Yeah, but to me, it really feels like what Sabine said. The time is now. What to me is the unique part of NANOSPRESSO is that we were forced, but luckily enough, we're forced to take the societal element seriously, because indeed it could have stuck with a technical concept and and we've proven and we are happy with the end result. That is where it normally ends. But now we were forced to think about also how it would end in society. How do regulators look at it? How do reimbursers look at this model? Are they willing to reimburse this at all? How do patients respond to this? Are they willing to be the first NS1 person to receive this new therapy where we say it's perfect, guaranteed safe? Well, are you convinced by the green light that emerges on the activation station? So that is, I think, really important. That's the lesson learned. There's a lot of momentum. All of the stakeholders seem to be aligned. Everybody sees the opportunity. Let's try to align all of our interests to make the best of it.
Wonderful. Well, thanks for those closing remarks. Thank you to you all, our speakers and panelists today, to Raymond Schiffelers, Sabine Fuchs, Olivia Lewis, Alexandre Ceccaldi, Alexandra Heumber. Thank you for joining us today. Thank you out there for being with us. I hope you enjoyed this session. Please visit the Frontiers in Science website to read the article we've talked about and the related content. You can continue the conversation on social media using the hashtag Frontiers Forum. But in the meantime, from me, from everybody here at Frontiers, enjoy the rest of your day. Thank you very much, and see you next time.
Thanks so much.
Bye-bye.
Bye-bye, everyone.
Bye-bye.